Welcome to the Fundamentals of R for Biologists webinar. So this webinar is going to just give a very, very brief overview of what R looks like, how do we actually use it, and what we're going to do is focus on using real biological data sets, and we are going to get them into R. We're going to very, very lightly clean them. We're going to figure out some summary statistics, and then we're going to visualize them using ggplot2. If you are wanting to know more about R, I do have some resources. Also, everything that we are going to be doing today, every single line of code that we are writing, I'm going to send out after the webinar. So if you're not on the email listserv for this webinar, there's a link down in the caption it'll also send you the intro material that we have here. So with that, I just want to get started immediately. So again, we can't cover absolutely everything. It's just going to be impossible to cover. I'm going to try and keep this at underneath an hour. We're going to see how well we can keep track of that. But let's get started in R. Now, if you have never worked in R, you don't even know what Rstudio is, that is totally okay. I will have to go through some of the fundamentals, the very, very fundamentals, very quick for you. So let's just get into R. So this is R. This is Rstudio. It may look a little different than your Rstudio. I use a dark theme because I have a little bit of light sensitivity. I'll actually show you how to do that at the very end of this course. If you don't have R and Rstudio installed on your computer, that is actually perfectly fine because there is a really cool website called Rstudio.cloud. It runs in your browser, you don't have to install anything, and it's free. So again, that's just Rstudio.cloud. So this is a really, really good place. I obviously use it every now and then when I just don't have access to my Rstudio, but that is an awesome place to start out if you don't want to install anything. I won't be covering how to install R in Rstudio in this particular webinar. However, a lot of these fundamentals, there's very basics of like how do you install it, what is R, how do you use it. I actually have an online course, and I'm not going to try to advertise it too much, but the first section is entirely free, and it covers how to install it, how to set the working directory, all of that stuff. But we will go through it very, very quickly right now. So if we're in Rstudio, right, we have a few main areas. And just to note, if you have questions throughout this webinar, ask them. Please ask them. I will try to respond to them live. Just put a big Q in front of it so I can see it. I won't be able to answer everything, and I'm going to leave space at the end for more thorough question answering. But just so you know, you can leave a question, just pop it right in chat. So here we have R. It is awesome. Whenever you first open up R, it's going to look like this. It's going to look completely blank, and we have a few different areas. We first have the console. The console is where we actually run code. So if I just want to run 2, let me make sure my keyboard is wireless and it died. If we went to 2 plus 2 and we run it, we see that we have 4. I'm actually going to zoom in on that just a little bit so we can see it a little better. So this is just, again, the console. This is where we run code. Now we also have the environment. The environment's up in the top right-hand side of the Rstudio environment. This is where we can view objects, view our data, and get a better idea of what we're working with. This will become more apparent when we actually load our data in. Finally, we have this lower section, which is down in the bottom right, which I'm covering up. This is a little bit of just a catch-all section. It includes a file browser. We have an area. This is where we plot things. Whenever we create a plot, this is where it goes. Also includes help documentation, which I highly encourage you use throughout R. Every single function in R has help documentation. And in general, this is just an area that we will use often. I'm going to move myself back into the corner. Whenever we work in R, you can totally just use the console. I don't recommend it. It's annoying. I would recommend creating scripts that you can run. So just by creating a new file up in the top left, we're just going to create an R script for this webinar. So in this R script, we can write code. So again, if I want to write any bit of code, let's do 3. Wow, I held shift, 3 plus 3. If I want to run this code, I can hit it with this run button, and it will be sent to my console, right? So whatever's in the source, if we run it, it gets sent to the console, and it's run. Throughout the course, I'm actually going to use the keyboard shortcut to run things, which is just Control-Enter. It's much faster and much easier. So if I'm saying I'm running code and you don't see me clicking the button, that's just because I'm hitting Control-Enter. So this is a very brief overview of just what is Rstudio. And this is where we are going to be working in R for this entire webinar and very likely throughout our entire course. Let me actually make this a little bit larger just so you can see it just a tiny bit better. All right. When we first start working in R, the very first thing we need to do is set a working directory. Working directories are like the bane of every newcomer to R's existence. Don't worry, we are going to break it down very, very simply. So a working directory is just a folder on your computer that allows you to actually see what you, that allows R to know where to work out of. So on my desktop, these are pictures, by the way, of red-eyed tree frogs eggs. I have a folder. I have this folder right here. It's titled Webinar. Inside of this folder, I have placed 2 data sets, okay? And these 2 data sets we are going to cover very, very quickly. So let's open up where I got these data sets from. The first data set is from Global Biodiversity Information Facility. This is an awesome website that you should probably be using. It has occurrence records for all life on Earth, and it's free and open source. I went through this and I found a data set that I could use for this webinar. I found one from the Ohio State University, their tetrapod division, their museum. This is their amphibian collection. So we see that there's about 5,000 occurrences of amphibians, largely based around Ohio and in the northeastern central-ish United States. There are records from elsewhere, but this is what we have. This is our data. I downloaded it, and now we have it inside of our folder. This is that working directory folder again, and I just named it osu_amphib.csv. So not birds, this one only includes amphibians, but you can get bird dataset from GBIF. Now, I also have another dataset in there. I titled it AmphiBIO. I also don't know if it's called AmphiBIO or AmphiBIO, but AmphiBIO is a global database for amphibian ecological traits. I've used this dataset a few times. This is the paper that introduces it, and all the authors definitely want to give them credit for this. It is an awesome dataset that links ecological traits to various amphibian species. And I have that inside of my working directory underneath this name, AmphiBIO_v1. Zooming in so you can see that a little bit more clear. Both of these are CSV files. So whenever we're in R, we actually want to get this data into R. It's great that it's in this folder, but R does not know that it's in this folder. There are thousands of folders on our computer, and we need to point to R where to go. We're going to do that, and we're going to do this in our script, so I'm going to keep this clean. So in our script up at the source, we're going to use the function setwd. This sets our working directory. And we can see with this little tooltip up here, we get a little preview of what this function needs. It needs a directory. What it really needs is the file path to the directory. Basically, we just need to tell R where to go. We just need to tell R where to find this folder. The easiest way to do that, so this folder, again, is on my desktop, okay? It's on my desktop, it's right here. I don't recommend keeping your folders on your desktop with all of your fancy data. Keep it somewhere secure, but I have it here. The quickest way to actually get that file path, just click into the address bar. This is all I do. Again, it's just here, up at the top, you have your little address bar. Click into it, that gets you your file path. I'm gonna copy this. I just copied it with Control-C, and then in RStudio, I'm just going to paste it inside of that setwd function. Again, this is very, very basics. I'm not going to cover this too much. I'm not going to spend too much time on this, but hey, working directories, everyone struggles with. So now that we have it inside of the setwd, we know where R can try to figure out where our data is placed. We just need to change those backslashes to forward slashes. This is just a little thing that we have to do in R. It's kind of annoying, but we have to do it. So we have it now set. This is our file path. It's telling R to go check my C drive, go into the users folder, the dil.j folder, the desktop folder, and then the webinar folder to work out of. If we run this, again, that gets sent down to our console right down here. We see that setwd has run. And the cool thing is we always know what working directory we're in by looking at the top of the console. Right here is our working directory. We see it has been set properly. Awesome. Now, let's actually get this data into R. The way we get data into R is by using the read suite of functions. Just like we want to get information from a book by reading it, we need to read the data into R. So what we do is I'm going to use the read.csv function. The reason I'm using that function is because our data sets are CSV files. They are .csv. That's that little file extension at the very end. Your computer might not be showing you file extensions. There are various settings you need to click to make sure you see them. But just so you know, these are all CSV files, so we're just going to use read.csv. And inside of read.csv, we just need to input the name of the file. Now, this is where it can get a little confusing, especially if you haven't been doing this a lot. Whenever we input the file name, it needs to be exactly what the file name looks like. So if there are capital letters, it needs to be included, lowercases need to be included, and we need to make sure we include that file extension. So if I want to load in the AmphiBIO dataset, I just need to type in AmphiBIO, bio is in all caps, _v1.csv. Now, if I run this, I'm going to go to this view, it will output into our console by default. The reason it's doing this is because we haven't assigned it to an object yet. We always want to assign our data to an object. That way we can actually manipulate it, we can get a view of it, and it's just much easier to work with. We do this by using an arrow. So these arrows are just telling us where data is going to. So we have the data that we are gathering from read.csv in that CSV file, and we want it to flow into some object. I'm just going to name our object AmphiBIO. You can name it whatever you want. That is totally fine. You don't have to name it anything. There's no real major requirements other than not using spaces. There are various characters you can't use, so just keep it simple. But if we run this code, what we're going to see is that it's now in our environment. So this is over on the right-hand side. And what we can see here is that the AmphiBIO dataset has various columns. We have 38 variables to work with and 67 observations. The datasets are not available for this webinar right now. I had to change the datasets a little bit right beforehand, but I will be sending them out after the webinar so you can follow through this entire pipeline step by step. I will very likely be sending links to the dataset instead of distributing it because it's not my data, it's someone else's data, but it's very easy to get access to it. So we have here now in our environment, again, we can see a preview of our data. If we actually want to see the full data set, we can click on this little table and it will show it in our source. Again, I'm zoomed in, so everything is going to be big. You can totally zoom out and make it easier. Here we can sort it, we can get a better idea of our data. You can't edit anything here, but hey, it's all here for you to see. Now, let's just load in the other data set really quickly. I'm not going to cover it again. So this is osu_amphib.csv. That's the name of the data set. So let's just assign it to an object called osu_occurrence records, because these are occurrence records of where species are found. And then we're going to do read.csv. And again, OSU, spelling it exactly the same way with punctuation, with the capital letters. And I'm going to run this code and you'll see it pop up in our environment. There we go. We now have the OSU occurrence record. So this is how we get data into R. There's a wide variety of read functions that are all suited for very specific types of data. And there are many different ways to load even a single CSV file in. I would encourage you to look through all of the read functions and figure it out. You can always Google what functions we need to load a specific data set into R. Now, if I want to access any of this data, we've assigned them to objects. These objects are data frames. Data frames are essentially Excel tables. That's the best way to think of them. If I want to access a particular column, let's say I want to access the AmphiBIO dataset and I want to get a particular column, right? Well, I just write out the name of the object and then I add in a $sign. This is how we get columns. So if I want to see all of the species in this AmphiBIO dataset, I just type out the name of the object, $species, run it, and then out of my console, I now see all the species. That's it. That's how we pull columns of data. We will also be using square brackets at some point. Make sure no typos. Square brackets are awesome. Our dataset is two-dimensional, so there's rows and columns. Inside of square brackets, we have a comma. To the left of the comma, is where we subset for rows. To the right of the comma is where we pull out columns. So if I want to get a specific column, right? Let's keep with this AmphiBIO dataset. And let's say I want to get the column species. I can type out the name, so I can do just like this. Again, it's to the right of the comma, indicating columns. I'm going to run this, and we get the exact same output down here. We get the species because we're pulling the same data. If I want to get a very particular row, I would then put that on the left-hand side of the comma. Usually with rows, we are going to be using a number. So if I want to get the first row, for example, we can do that. My output is going to look hideous because we're zoomed in. So just rest assured, this is a row of data. That just looks terrible. But let's do a better example. So let's say I want to get the first 10 species found in my AmphiBIO dataset. I can do 1:10. This just says go from 1 to 10. And if we run this, we'll actually see the first 10 species in that data set. So this is a very, very, very simple, very quick intro to just subsetting and pulling data out there. Let's install a package, okay? Let's use a package that is going to make our life a lot easier. And yes, this video will be accessible after this live session, and I will be including chapter marks so you know exactly where to go for each section. One huge benefit of R, being R, is that it is free and open source. This means that anyone can download it and create their own custom functions. You can as well. You can create your own custom functions, you can get it uploaded to the CRAN repository, which is where R is housed, and you can send those packages to people so that they can use your functions. One of the best packages out there is Tidyverse, and I highly, highly recommend every single person learn R with Tidyverse, and that's what we're going to be doing today. Tidyverse is technically multiple packages inside of one package, but they are specifically suited for data manipulation, data structure, and data visualization. You may have heard of ggplot2 and dplyr and tidyr. Those are all found in Tidyverse. So to install a package, we just do install.packages, and then we type out the name of the package. So in this case, Tidyverse. Here, we're just installing the package. This is sort of like buying a book and putting it on the bookshelf for later. We don't have access to the functions yet, but we can if we want to. So I'm going to install it. I'm going to run this. I already have this package, and it's actually loaded already, so I'm not going to restart R. Usually a whole bunch of text is going to pop into your console, and it's going to say, oh, it's installing everything, but mine's already installed, so it's not showing that. Once we have the package installed, we need to take that book off the bookshelf and actually get the information contained within it, right? And the way we do that is with the library function. So library, and now we just input the name of the package. Note that we don't need to use the quotation marks anymore for this. If we run this code, it will now run library, and we now have all of the functions from the Tidyverse package available to us in R. I mentioned that we have dplyr, for example. So if I type in dplyr and do a double colon, we now see every single function from that dplyr package. If you do this with Tidyverse, it won't show anything because Tidyverse is loading all these other packages for you. So now we have some of the best functions in the world for manipulating, cleaning, and visualizing our data. One of the best elements of Tidyverse is the pipe. The pipe is a very simple but extremely powerful bit of code. All it is just a %, this little greater than less than symbol, and the % again. And yeah, just to note, if you are using the Tidyverse package, you don't need to install or download any packages. You don't need to like go onto a folder and go to tidyverse.org and download it. Everything that you need to do to get Tidyverse inside of RStudio is just this line of code. That's it. So with this pipe, the way it works is that we have information on the left-hand side of the pipe, and that flows into the information on the right-hand side of the pipe. Just like with a pipe in real life, water or some liquid or some gas will flow from one side of the pipe to the other side of the pipe. That's all this is doing here. What we can do is we can then start feeding in data into either side of our pipe. So for example, Let's see. It seems, let's do a very simple function that is from Tidyverse, okay? We want to clean up our data sets. These data sets are huge, okay? We have 5,000 observations, almost 7,000 observations. We have, over, we have 50 variables, 50 columns. This is a lot of data to go through. What we can do is we can start by using our first data set. This is our initial data set. This is what our input into our pipe is going to be. It's on the left-hand side of our pipe. On the right-hand side, let's use a function. Okay, the first function I want to introduce to you from Tidyverse is select. What select does is it selects columns from our data set. So let's say we want to pull out a column. Let's say we have AmphiBIO and we want to pull out species. Okay, awesome. Or I'm sorry, we're using the occurrence records. We will also pull out species. So let's select species. That's it. Now that worked. So of course, this is the exact same thing as just doing OSU occurrence $species. Of course, this is the exact same output. We have species outputting into our console. This is just to illustrate how pipes work. So the information from OSU occurrence is being fed into the select function, and then the select function is asking us, hey, what columns do you want to pull? Well, I want to pull the species column. So that's how the pipeline works. As we build out our pipeline, the pipes will start to make a little bit more sense, and I will really be focusing on how they work. So let's get a few other records from this occurrence data set. I want to get a little bit more information about the taxonomy, so let's get order, let's get the family, and let's get the genus. These are all separated by commas, so this is going to pull those 4 columns right there. If we run the data and we look at our console, we see that we have those 4 columns now. We don't have all 50 columns, we just have these 4. I'll have to scroll to the top so you can see the headers, so order, family, genus, and species. This is occurrence records, and we would want to know where those occurrences were found. So let's add in decimal latitude and decimal longitude. These are the column names. It's nice that also whenever we're working in Tidyverse, the nice thing is with pipes, you get auto-completed a lot. So it makes your life a heck of a lot easier. So again, if we run this in our console, it's an abbreviated output. So we have the first 4 columns here, and then down here, these are the next few columns, so decimal latitude, decimal longitude. Super, super easy. Now I'm going to clean this up just a tiny bit, so let's make this a little bit longer. And the cool thing is with pipes is that if you have a pipe at the end of a line, you can start your next line right below it. So this makes it super easy to code. Let's add in one more pipe, okay? I want to do one more thing. Because if we look at this data set, right, let's scroll up to the top. We have this species column, right? Well, we want to know the species. But if we scroll through it, we'll see that we have a few blank values. These are not included in the species column. They're blank for whatever reason. Most likely, these were records that could only be identified down to the genus or to the family. So for example, this one is a hyla. It's one of the tree frogs found here. But they didn't know what species it was. There's a few others in this data set. I believe we have a preview of Arana, which is like your true frogs. And again, it's blank. Well, we want to filter those out, right? We want to just remove those. We're just not interested in records that don't have the species. Well, we can filter them out with filter. This is another function from dplyr, which is again from Tidyverse. As a reminder, we have pipes at the end of each of our lines of code. So let's use a filter. Let's say we want to pull out species where it is not blank, okay? So we specified the column that we want to do some type of logic on. We want to check true, false, is this our species there, our species not there? This little code right here says not equal to. The exclamation point means not. If we wanted to find values that were only blank, we would use 2 equal signs. And yeah, just using 2 quotation marks just means blank. This is different than NA values, which we'll cover in a bit, but these are just truly blank values. So if we run this, right now this is showing the equals blank. We are now getting the values that are only where species are missing. So we can actually see there's quite a few records that are missing the species, right? There's this species column right here and nothing below it. I think a lot of people get confused with filter. Filter does not mean filter out. It means filtering in. right? So we want to only choose records that are equal to what our filter is specifying. So in this case, we want to choose records where species are not blank. So if we run this now and we look at our output, we see that if we went through every single one of these thousands and thousands of observations, we would see that all of the species are represented and there's no blank values. And now this is the cool part, and it's just perfect because this answers a question in the chat. You can assign the output from tidied pipelines to a new object. You can do it just like we do up here with an arrow and then the name of the object. You can 100% add it at the start of your pipeline if you wish. So we could do a new data set here, OSU occurrence clean, and this will totally run. What I prefer to do for pipelines is actually put it at the end of the pipeline. So all we're going to do is we're going to put the arrow going the other way, because remember, it's information flowing from one side of the arrow to the other. And I'm just going to shamelessly copy and paste that into R. This makes more sense to me because we are starting with a pipeline. We're starting with our raw data set. We are selecting columns. We are filtering out blank species. And then we are assigning it at the end of our pipeline to a new object. And in fact, right now we haven't even assigned it yet. If we run this code, we now have that over in our environment right here, OSU occurrence clean. We see that we have only 6 variables, so 6 columns because we selected 6. And then we filtered out, looks like we removed almost 200 records where species were blank. I'm going to say this is it for cleaning. I will say, especially with GBIF data, the Biodiversity Information Facility, you need to clean your data set really, really well. This is not sufficient cleaning to use this data for a publication, so just make sure you clean it more thoroughly. We will probably talk about that sometime in the future. I use this every single day. Let's now clean the AmphiBIO dataset really quickly. I'm not going to spend as much time on it. We're going to select columns again. We have 38 columns. We don't need all of them. Let's get the species column. We're going to ignore all of the order, family, genus stuff, so we'll just type out species. Again, this species has a capital S, so we need to make sure we include it. And let's actually include a couple different variables that relate to the lifestyle of amphibians, okay? Here we have a fossorial, terrestrial, aquatic, arboreal. That's what these mean. I'm going to just pull terrestrial, so TER, make sure it's separated by a comma, and arboreal. Okay, that's all we need. And just for reference, whenever we output something from Tidyverse, this is going to output as a data frame by default. There are different functions that may change your data and manipulate it in some way, but usually if you feed in a data frame, it's going to output as a data frame as well. So we're pulling out these select functions. Let's also pull out the body mass function. So there is a body mass variable. So we have body mass in grams. Let's also pull body size in millimeters. So again, if we select it, it looks like we have a typo. Yep, I included a period instead of a comma. Run that again, it works. Typos happen. Typos are going to happen. Embrace the typos. I wouldn't say learn to love them, it's impossible to love them, but learn to accept them for who they are. So now we have an abbreviated data set of AmphiBIO, right? We have the species, we have if they're terrestrial or arboreal, represented by a 1 if they exhibit that type of life history trait. And we have the average body mass and body size for each particular species. So yeah, all represented here. Now I want to introduce a new function. We're going to introduce mutate, because mutate is so useful. It's another function from Tidyverse that just changes your whole life. So mutate allows us to mutate data. It allows us to create a new column in our data set or overwrite a column in our data set and create a new one, effectively create new data. So let's say I wanted to convert body size in millimeters to body size in centimeters. We'll make a new column for that. We'll call it body size cm. And we need to add the equal sign. We want to say what that is equal to. Well, we want to convert body size in millimeters to centimeters. So how do we convert millimeters to centimeters? Well, body size underscore mm, and we just divide it by 10. That's it. This will convert the millimeters column to centimeters just by using mutate. So if we run this, it's going to add it at the end of our data set. So let me actually let me make this a little bit wider so it's all on one, one view makes it a lot easier for us. So here we have that original body size millimeter column. Right next to it, we have the new centimeter column. This is a very, very easy way of manipulating data. We could have overwrote the body size millimeter column if we wanted to. We would just have to input that here. However, if we did that, then we would have values that are now converted to centimeters in the millimeters column, and that's just a mistake. So again, we're doing very, very simple cleaning here. There's a lot of different functions you can use. I will send out some materials that will cover all of this, and also there are links to a full textbook which includes many other functions, which is 100% free. But let's now just call this AmphiBIO _ clean. So that's going to be our new data set. Just so you can see it again, just so we're cementing knowledge, so we're understanding how everything works. We have the original AmphiBIO dataset that is raw. We are selecting a few columns by going through a pipeline. And then the next step is we're going to take all of this data here, this abbreviated dataset with only a few columns, and we're going to mutate it, and we're going to add a new column that just converts centimeters to millimeters. And then finally, we're going to assign it to the AmphiBIO clean dataset right now. Look at that. We have it over in our environment. Awesome. Now, this is where I want to have some fun, because one of my favorite functions in R is merge. Often working with multiple datasets, it's kind of tricky. Often in biology, we need to combine datasets from multiple sources. We may have multiple surveys. We may have one dataset that needs to be paired with another dataset. This is actually an example of two datasets that would be really, really useful if they were paired together. So that way we're not working in two datasets, we can just work with one. So again, I'm going to use that merge function I mentioned. Now merge just merges two data sets together. Let's make sure this is front and center in our screen. I'm going to zoom in on it just a little bit. So what we need to input into merge are a few different arguments. First, we need to input the data sets we actually want to merge, okay? So let's start with OSU occurrence clean. Again, we want to feed the clean data set in, not the raw data set. And then we want to feed an AmphiBIO clean. Now we need to tell R how we need to merge these data together. How are we going to merge them together so that we have one beautiful data set? The way we merge is by looking at a column that is shared between both data sets. If you remember, we pulled the species data for both of these data sets. So it makes sense that we would merge on the species column. That way, all of the values from AmphiBIO clean, all those life history traits that we extracted, are going to be paired with the occurrence records from our GBIF data. So what we need to do is specify it with by. What are we merging by? Because our species column is actually spelled differently in both, one has a lowercase s, one has an uppercase s. Yes, we could just change the names. I'm not going to because this proves a point. We can specify each data set individually in merge with by.x and by.y. The x data set is our first data set, and our y data set is our second data set. This is usually how it usually the standard for R. Not all functions follow it, but usually the first data will be the x, usually the second will be the y. So in by.x, what do we want to filter? What do we want to combine things by? Well, we want to combine them by species. So we'll add that. And then I'll show you a little trick here. Just like how with pipes, if you have it at the end of a line, you can just put the next set of functions on the next line within a single function. So this is a single function encapsulated by these parentheses. If there's a comma, you can actually put this on a new line. So I'm going to do that just to make this so much easier to read. This is all working. Again, the comma needs to be at the end of each line, though. So by.y also equal to species. Let's run this. And then right now it's just outputting to the console, but we see our data is merged. We have the species, the order, family, genus, we have the latitude, longitude, and then next to it we have these life history traits. We have body mass, we have body size, we have everything here. So let's assign this to a new data object. We're going to call it df merge. Df is usually what I like to call my data names. So right now this is over in our environment, df merge. And we see that we have 4,600 observations of 11 variables. The merge function does automatically remove records. So if there were values that were not found in one or the other, so say there was a species in the occurrence data set that isn't found in AmphiBIO, or vice versa, it just gets removed. You can specify to not remove them using this all argument. So if it's like all .x, so we want to keep all the, oops, sorry, you're not seeing that. So all.x, we want to keep all of the records from the x data, that OSU occurrence. We could set it equal to true or false. I'm not going to, but just so you know, that exists. Now we have the merged data set. We finally cleaned it. We have our data in a format that we can actually use it in. Let's actually calculate some statistics. Let's actually figure out some stuff, okay? So let's create a new pipeline. Instead of a pipeline for cleaning, let's do a pipeline for analyzing and summarizing our data. Now, I will be very let's introduce a quick function here. Within our data, if we look at our DF merge data set, again, I just click that little table button next to it on the environment. If we look at body mass and body size, and you'll see a lot of these values they're not necessarily, they are duplicated, but that's because there's a lot of records of like Acris crepitans, the cricket frogs. So it's going to be there a lot. But if we scroll through body size, if we scroll through body mass, what we're going to find, excuse me, it seems like there's a plane flying over right now. What we're going to find are a lot of NA values, okay? So of course I'm trying to find one now and it's just not pulling out. But let's look at the original data set from AmphiBIO. So we have a lot of NA values in our data set, and this makes it very difficult to run metrics such as mean. If we try to get the mean of this column right now, it's actually going to return NA. So let's first have a filter that removes those NA values. There are many different ways to handle NAs. We're going to filter them out. We're just going to outright remove them. So again, we're going to use that exclamation point, means not. We're going to zoom in on this so you can see it just a little bit better. And we're going to use a function called is.na. Now we want to choose which column we want to go through and figure out which one do we want to remove the NA values from. So let's say we want to get the average body mass, okay? So we just type in body mass G, and this just removes the NA values, okay? Now let's add in another pipe and another function. The most useful function you can use in R is called summarize. You can spell it with a S or a Z. It's like a, like American English versus British English thing. But either way works. Summarize is often needed to actually summarize our data in some meaningful way. So if I want to get the mean of body mass, I first need to add summarize and then do mean and then body mass G. Make sure I spell it correctly. So let's figure out the mean body mass for all of our amphibians that were found from the OSU occurrence dataset. It actually went to the console. So 86.5. That's the mean body mass for all of these amphibian occurrence records. Now, if you are really in tune with the data, you may notice that this is actually not the correct mean. There are some issues because the data is duplicated. So records of species that are found often are going to influence this mean versus species that are rare are not going to have as much influence on this mean. So just being very forward about how we're using our data. But maybe we want to pull out the body mass for each of the species, right? We want to know how to do that. There is a really, really useful function in Tidyverse called group by. It allows us to group our variables or group our data according to some variable. And I'm just going to add it into our pipeline. I'm just going to directly add it in. Right after we filter our data, let's now group our data. So what do we want to group it by? Well, let's say we want to get just the average body mass for each family, right? Because remember, we included that family data. So all we did was we added group by. How does this change our output? Well, it has grouped our data according to family and then calculated the mean for each of those groups. Now we see that the ambistomatids have an average mass of 31, all those mole salamanders. We see our tree frogs have an average mass of 3 grams. We see our sirens have an average mass of 1,000 grams, a kilogram. So all of this was accomplished just with a very, very simple pipeline. There wasn't a whole lot we can do. And if we want to change it, if we want to figure out something else, it's so easy. We just go into family. Let's say we want to figure out genus. Boom. We now have the output for different genera. So it is super, super easy to accomplish anything you want just by using filters, group bys, and summarize. Now I want to introduce one other function that is really, really useful, and that is count. So what I'm going to do is I'm going to, we'll just start it over again. So df merge, and then let's add a pipe, and then let's group it by species, okay? Group by species. In this case, we're not worrying about body mass, so we don't need to filter out NAs. We already removed the blank species. So let's add another pipe, and let's add count. What count is going to do is it's going to count the number of records we have for each group. So if we have 5,000 records of amphibian observations, obviously there's only, I believe there's only 140 species or so, but there's all those records. Now we can see how many times each of those species appears in our data set. We see Acris crepitans, or cricket frogs, were found 165 times. We see lots of different Ambystoma up at the start. Were found once, 13 times, 41 times, 83 times. Count is really, really useful for just getting an idea of your data. Now, these are just very simple summary statistics, okay? There are a wide variety of different functions we can use in Tidyverse. These are probably the most commonly used and probably the most helpful. Again, filter, group by, summarize, the count function, all of these can accomplish honestly 90% of what many ecology-focused biologists really need to use. You can also plug in analyses. So I've actually done this where I've grouped by some group and then ran t-tests over every single group compared to some control population. So that's an example of how you could use it for an analysis, for example. I'm not covering analyses here just because there's so many different ones we could do. That's going to be a whole other course. I just want to give you the tools so that you can work in R. And now let's cover one of the biggest beasts in R that everyone struggles with. I still struggle with. I am always Googling things, looking up cheat sheets, and you should too. These are not closed book exams whenever you are coding in R. Just Google it. It's totally fine. Let's cover ggplot2. So ggplot is the most widely used data visualization package in R. It is used by so many people. If you are on the R Twitter, you are seeing people constantly sharing their really cool ggplots. And if you're like me, often you're looking at it and you're wondering how the hell are they doing that. So let's break it down. In ggplot is our initial function. Okay, we want some data to go into our ggplot. Let's just use our merge data set, right? With ggplots, there are three components we need to be aware of. We need to be aware of our data, so what data is going into it, in this case, our df merge data set. We need to be aware of various aesthetics. So aesthetics are what we see, but within the ggplot function, the aesthetics are used to map data to what we see, right? So if we have this aesthetics argument, which is just AES, again, that X and Y that we were talking about, the first data set will be X, second will be Y. Right now, we want to figure out, hey, what values do we want to put on our x-axis? What values do we want to put on our y-axis, right? What data do we want to map to that x and to that y? So let's first map our x-axis. Let's say I want to put body size on the x-axis. Well, that's easy. We just put body size. Body size mm. Easy. And then on the y-axis, we put the variable that we want to put on the y-axis. Let's just do body. Let's do mass. Let's do mass in grams. If we run this plot right now, okay, I'm gonna make sure. I'm gonna actually move myself. Apologies for the mess at the moment. I just want to make sure that this is shown properly. If we run that ggplot where it's taking from df merge and we're assigning aesthetics, we're assigning body size to the x-axis, we're assigning the body weight, the body mass to the y-axis. If we run this, we have nothing plotted. However, what's going on is that we can see how our data has been mapped to those various aesthetics. we see that body size is now on our x-axis. And we see the x-axis has already been custom, has been put together so that it is the correct scale. It's already using that data to create the x-axis. And then the same thing on the y-axis. We have body mass in grams, and it's already using the scale based on that body mass data. But nothing is plotted, okay? That's where the third element comes in, geometric layers. Our, ggplot uses geometric layers constantly. I'm gonna put myself in the center of the screen so we can see everything all at once. So if you've already tried plotting in ggplot, you may have noticed we have geom. Geom stands for geometric layers. And we see here, these are all the different ways that we can plot data. Essentially what we've done with this first line is we've just told R what data to use and where does that data go. But it doesn't know how to actually visualize that data. It doesn't know if you want line plots. It doesn't know if you want a bar chart. It doesn't know if you want a scatter plot. That's where the geometric layers come in. So if I want a scatter plot, I'm going to use geom point. This creates a scatter plot based on our data. That's it. If I wanted to change this to a line plot, not actually sure if that'll work with this data set, but it would make lines. If I want to make a box plot, there's geom box plot. Now, for the sake of the rest of this section, okay, I'm going to change this scale because we have data that is way outliers. I'm pretty sure these are the sirens, actually, because they're quite large. What I'm going to do is I'm just going to change the scale. I'm not going to cover this too in depth because we could spend a whole long amount of time on adjusting every single element of R. But let's change both of our scales. We're just going to use continuous scales because this is continuous data. And we're just going to transform it. We use the trans argument to transform it into something. Let's log transform it. We'll do log2. So I'm going to change both of our scales. Again, this is something I'm going to gloss over kind of quickly. But you're probably going to want to change the scales of your data at some point. And this is how you do it. You just do scale X, scale Y. Now we see our data is scaled according to a log scale. So that it just makes it look a lot easier. It's a lot easier for us to actually work with. And in fact, I just realized the y-axis has not been changed. So now it's exactly what I wanted it to look like. Scale X and scale Y continuous, and the transformation is just log. Awesome. Now let's actually start customizing this a little bit. So I'm going to move geom point to the end, just because I think it looks nicer that way. Boom. Oh, I just deleted it. I didn't. Okay, geom point. Cool. So it works. The order of this matters in terms of adding on layers. So let's say I wanted to add a line chart. I wanted to have a line of best fit that actually describes this data well. So geom smooth is what we're going to use. This just creates a line of best fit. This one is a little bit more complex. It's using a more complex data set, so we see that it dips down there. However, this is going to showcase our different layers really, really well. So first, what I'm going to do is I'm going to change the size of our points. Let me just do that with size. I'm going to set them equal to 5. These are going to make them huge, okay? And this is by design. ggplot works on layers, okay? Right now, the geom point layer is rendered onto our plot before the geom smooth layer because it's rendering top to bottom, right? And we can see that on our plot. We see the line is over our points. Let's just reverse them. Let's put geom smooth first. Let's make sure that renders on our plot first, and then geom point. What's going to happen? The line is now behind our points. So this is something just to keep in mind whenever you're working in ggplot, is that layers are a thing. Layers do influence how our plots work, and we can actually use this to create multiple different graphs on one thing. If we wanted to add another plot on top of this, we could. If we wanted to add labels, we absolutely can. So like I mentioned, there are a lot of different size. There is shape as well. So if I wanted to make the shape triangles, we could do that. These are going to change the points that we have. And let's also just change the color. Let's just make it red. You know, screw it. Let's make these giant triangle red triangles. Cool. We can do that with these arguments here. Size, shape, color, red. And what we can then do is we can use aesthetics. We can actually map data to these variables here. We can map data to color, we can map data to shape, we can map data to size, okay? And we're going to do that with this aesthetic argument again. Now I'm going to actually simplify this code massively, make it look very simple. So I'm going to remove the smooth, we don't need it right now, let's just simplify. And I'm actually going to assign our ggplot to an object called gg. You can name it whatever you want. I'm going to name it gg. This actually creates an object in our environment that contains all of this information. So now, if I want to do a very simple, if I want to make a geom point, I can just type in gg plus the next layer. Run this, we have the same thing. We have the axes scaled, we have the same data, it's still mapped, and we remove that line. So that's no longer there. But now let's just remove all this. Let's just keep it simple. Let's plot it like this. Let's say we have all these points, right? These are all different amphibians, but it's not really descriptive. I mean, we don't know what family of, we don't even know what species are up here. We don't know what family is down here. Let's color our data according to family. Now, if you are using data to visualize something in R, if you're using it to change colors, if you're using it to set the size according to some data, it needs to be within aesthetics. It can't be outside, it has to be within. So let's say we want to color it according to, let's do family, right? Let's actually make the size 3. What we see is that our data is now colored according to family. Now, yes, this looks hideous. This is a massive thing. We are not covering how to make pretty ggplots in this section. We're just covering all the basic components. But now we can see that generally our hylids and our eleutherodactylids and our leptodactylids are kind of small, and they're all kind of down here in body size, body mass. We see that we have the crowgasters sort of in this general area. This is a really useful way to set some type of information according to variables. We could also use this size argument. Let's say we want to make it to where our points are according to body size. So let's set it. Again, we removed it. It needs to be a capital B. We removed it outside of aesthetics. We put it inside of aesthetics because we are using data to set this. So now the size of these points is according to the body map, to the body size. So as the body size increases, so too do the points. Now, this is a super very brief intro to ggplot, and I want to leave you with one extra function, okay? So let's say your advisor comes in and you show him this plot. Let's say you didn't color by family because you realize that there's too many plots and it looks kind of hideous. And they come in and they are wildly disappointed with what you're doing. I'm trying to make this webinar more relatable for people. Your PI is upset because they don't understand this plot at all and they don't know what's going on. They actually want to see how the different families actually look, and they want to see it way more clearly. What we can do is we can facet our ggplot. There's a couple different faceting functions. We're going to use facet wrap for this. The differences are nuanced between the two. Let's say I want to see by family. If we add this little tilde, this is a squiggly line, as a lot of people call it. The tilde is at the top left of your keyboard. Definitely learn it. If you're going to be using R, you're going to use the tilde a lot. Let's say I want to look at it by, like we just said, family. Now, when I run this code, I'm going to make this plot bigger. What it's going to do is it's going to take the family column. It's going to figure out all the unique values of the family. So, there's 5,000 observations, but there's maybe, 40 families. And it's going to segment them for you and plot each one of them individually on the same plot. So obviously our text is way too big. We could change that out. I'm going to move myself out of the way. But we see now we have a plot just for the hylids. We have a plot just for the raynids down here. And if we include the information, if we include some aesthetics up here, it's going to go down into our facet as well. So let's change this size. Let's actually just change it back to color, and let's color by family as well, right? This is just to illustrate the point. Each one is going to be colored individually, right? Because we're already separating them by family. So all the hylids are one color. We do have some blank values here, which totally does happen. Don't worry about it. Again, not making clean looking graphs. If we want to subset, if we want to facet according to some other variable, let's say, let's do it by order so we can actually see salamanders versus frogs, right? So let's do it by order. Order, yep. Anura, Chordata, frogs, salamanders. And we can see that, hey, look, it's still colored according to family. All the families have their own unique color, and we can very easily see different trends for frogs versus salamanders. This is probably one of the most useful functions within R, within ggplot, is the facet wrapping. Okay, now, I had to make some changes for this presentation. I unfortunately did not have the time. We're already at the hour mark. I did not have the time to go through if-else statements and for loops, unfortunately. I really wanted to get to that, but we didn't have the time. I will be sending out material after this webinar that is going to show everything we wrote here. And I'm going to include detailed notes as well. So I'm going to talk exactly about what the read.csv function is, how it works. I want to make sure this is extremely useful for you. If you had an RSVP email, if you got the intro material that I sent out, you will automatically get this, hopefully tomorrow, if all things go well. And I am going to try to have a part 2 where we actually go into the next steps where we do for loops and if-else statements. Now, you've reached the end of this, and I just ask to indulge. If you have questions, please ask them. But I want to show you two things. This is the webinar landing page. So if you did not get the intro material, a link to this page is actually down in the caption. And if you just get your name and email, I can send you a 30-page PDF that covers all of the material from this webinar, or at least as much as I could. And then I will also send you all of the material that we covered today. Further, all of this material, all the material I sent here is from this ebook, which is actually just now launched. I just got it ready last night. This is about 150 pages of Fundamentals of R for Biologists, and it is actually tied to my course. And this webinar will be available afterwards. I will be editing it down and making sure it's available for all of time. But I do have a course that covers all of these materials. Again, the first section is 100% free. Every single lesson, there's 73 lessons, if I remember right, all covering various aspects of what we've covered here. It includes pipes, it includes all of the written material, as well as video lessons for every single one. So if you don't know the working directory, if like you struggle with working directories, because I do too, sometimes I get really confused, you can go to the working directory lesson. There's a video and the full written text. This is actually the exact same text as the textbook, and we have all of the lessons here. Further, there are quizzes. These are open book quizzes just to cement your knowledge, as well as mastery checks, which are sort of like mini projects. And I will also be including this webinar, this material, and the full workup of what we are doing in this course as a case study. This is going to be for free totally. The course, again, only the first section is free, just so you can see if this is something for you, if this learning style works for you. But if you actually look inside of the caption, there is a coupon code for this course. So it is just webinar 22. It'll give you 20% off on the course. And this thing will live forever. You have unlimited access. And yeah, so I have some time. If there's any questions that you think I didn't answer, you want me to cover something again, please ask. This section will actually not be probably on the... No, I'll keep it in the recap. We'll keep it in the recap. But yeah, I hope this was really, really useful for everyone. I have been working on this course and webinar for about 3 months now, so just wanted to make sure that it was accessible for everyone. I am working on also translating the textbook into Spanish. I need to find a good source to do that. That isn't going to break my bank, because I just know sometimes, oftentimes, hearing explanations in your mother tongue is really useful. But yes, this is how we analyze biological data sets. This was a very, very quick, I think very dense overview. The recap